Hi, everyone. Great to see you here. We have about 5 minutes, actually 4 minutes to get started. I'll be the moderator for this room. And for those who are presenting, please be sure to add yourself to the moderator queue. I can see that Tyler, Sammith, and Ishaan have already done that. We are also looking for Jacob and Tanisha to do the same. Okay, Jacob has just done the same. So we are looking for, Tanisha. Tanisha, are you here? All right, I'll also start the chat here in the session chat. Hi, everyone. Just so you know, if you click on the top right session, then the chat will be specific to this session. If you click on the event tab and there you kind of write anything in the chat, that will be seen by everyone across all of the sessions. So say hi here in the chat, in the session chat. And also, I believe, yes, I'd like to welcome Andrew Smith, our additional judge here. I'll also be judging, and Andrew will also be judging the talks. So hi, Andrew. Please add yourself as a presenter for a moment so I can give you just a minute to introduce yourself. Hi, Andrew. Andrew, please, I'd love to introduce you with your camera on if you are ready for that. If not, that's also fine. But Andrew says hello to everyone in the chat. So Andrew is trying to share. So Andrew, I cannot see you in the moderation panel where I would need you to be added in order to share your screen. We'll wait just one more minute for Andrew. In any case, in the meantime, I'd like to ask Tanisha to join the moderation panel. Tanisha, are you here? Tanisha? Got it. Okay, so we have 1 more minute left. So I'll just quickly recap how we are going to run this. So each talk will be 10 minutes in length. We will start with Ishaan. After Ishaan comes Sammith, and then Jacob, and then Tyler. And hopefully in the meantime, Tanisha will also show up. Tanisha will be the 5th speaker today. And you have 10 minutes to talk. And after 10 minutes, well, after 6 minutes, I'll rejoin without my video on as the moderator in the moderation panel. That will be an indication for those of you speaking that you only have 2 minutes left. And after 2 minutes, we'll have 2 minutes, and after the 8 minutes are up, we'll have 2 more minutes of questions. So let's get started then. So Andrew, let me just, so Andrew is introducing himself in the chat. So Andrew is a marketing PhD student at UPenn, and Andrew will be moderating, we'll be judging the talks today. And just a few more words about Andrew. Andrew's research centers on interpersonal dynamics, and that's a very varied research topic. He has done research on help seeking, on delegation services, on emotions, and brand attachment. And I'm sure Andrew will be happy to give you more details about his experience at the Wharton School of Business at UPenn if you reach out to him. With that, let's get started. And I'll be leaving the stage. And as I mentioned, the first talk will be by Ishaan Bhargava, a high school student at Greenwood High International School from Bangalore, Karnataka, from India. Ishaan, please join the stage. Ishaan?
Yeah, hi, can you hear me?
Yes, wonderful. Ishaan, so please share your screen and I'm leaving the stage.
Yeah. Can you see my screen? Okay, so I guess I'll start. Uh, Hello everyone, I'm Ishaan Bhargava, and today I will be presenting the research project I did with my mentor Keith Shipper through Polygence on the interrelationship between COVID-19 outcomes, pollution, and mobility. So, a little bit about myself. I'm 17 years old and I stay in Bangalore, India. Some of my interests include playing badminton and playing the guitar. I have an interest in the environment as well as technology, and in the future, I hope to combine these two interests. So, before we get into the presentation, I want to give a small roadmap of what all we will be covering. So, firstly, I'll be discussing on how I actually arrived to my problem statement. I will then introduce the variables of interest and give a small background about them. I just want to briefly discuss where I got my data. And next, I'll be telling you all about how I actually conducted my analysis and then going over the results and how I interpreted them. Finally, I'll be discussing the major findings and their implications on the real world. So, I wanted to start off by showing you all two world maps. The one on the left is a pollution map, and the one on the right is a COVID-19 per 100,000 of the population map. So, as I put these two maps together, I saw a lot of similarities in places. Countries such as Russia, countries in Europe, and countries in Africa had similar rates of pollution and COVID cases. And this really got me thinking on whether there could be a relationship between COVID-19 and pollution. So, I wanted to explore a region I was familiar with, and that's why I looked more into India. You can see the pollution map of India on the left, and you can see that there is a lot of pollution concentrated in the north, especially around the Delhi area. New Delhi is the capital of India. So I personally knew that Delhi was severely impacted by the COVID outbreak, reaching up to 24,000 cases per day. And that's why I felt that Delhi would be an interesting region to study. So, to summarize again, Delhi was chosen as my region of study because a metropolitan city like this, with high amounts of pollution and population, would see a more noticeable and drastic effect, if any. So, this gave rise to my research question, which was to investigate the relationship between air pollution and COVID 19 infection, as well as to assess the modulatory role of mobility on this association. So, now a small background to my variables. The air pollutants I used in my analysis were particulate matter, which is basically the term for a mixture of solid particles and liquid droplets found in the air. I also included ozone, carbon monoxide, which is formed by burning fossil fuels like natural gas, petrol, coal, and oil. I included nitrogen dioxide, which can be further categorized into nitrogen oxides as a whole, which encompasses nitrogen oxide as well. The last few pollutants are sulfur dioxide found in burning of fossil fuels and volatile organic compounds such as benzene, toluene, and xylene. So moving on to mobility. In my research question, you may have been wondering what mobility is and why was it included. Mobility essentially describes movement trends and how people's movement changed throughout the pandemic. And these can widely be categorized into six main types, which you can see. Places including restaurants, supermarkets, national parks, train stations, workplaces, and residences. So the graph below just gives a timeline of how the mobility in India changed over time. So I took my COVID data from the Ministry of Health and Welfare, my pollution data from the Central Pollution Control Board of India, and the mobility data from the Google COVID-19 Community Mobility Report. My date range for the analysis was from the March of 2020 to July of 2021. I chose this because I felt that it encompassed both major COVID waves in India properly, and also my data sets were limited to these dates. So now going on to the analysis. My analysis consisted of two major steps. The first, building a correlation matrix. A correlation matrix is a table showing correlation coefficients between variables. The correlation coefficient is a statistical measure of the strength of the relationship between the relative movements of two variables. Building a correlation matrix between all three major variables helped me get a sense of how each of my variables was related to one another, and also helped me eliminate very similar variables to prevent my model from getting overburdened. Most of my analysis was done using a negative binomial generalized linear model. Generalized linear models estimate regression models for outcomes following exponential distributions. The negative binomial distribution, which you can see on the right, fitted my data best, and I used this model to get my results. And here are my results. So I created two models, one for the daily cases, the one on the left, and one for the daily deaths, the one on the right. Essentially, each value tells us that for every 1 unit increase in the independent variable, so independent variables are the ones which are on the x-axis, there will be an x% increase or decrease in cases or deaths. For example, if you take residential mobility, the incidence rate ratio tells us that for every 1% increase in residential mobility, there could be up to an 8% decrease in cases and 6% decrease in deaths. Now, in this model, I found three variables that really stood out to me. Those were carbon monoxide, sulfur dioxide, and residential mobility. Going into detail, for carbon monoxide, a 1 microgram per meter cube increase could cause up to a 23% increase in cases and 25% increase in deaths. Now, this was a very, very strong relationship. And after doing research, I found that there are strong links between carbon monoxide and COVID onset lung diseases like acute respiratory distress syndrome, known as ARDS. So, what carbon monoxide does is that it affects human health by impairing the ability of the blood to bring oxygen to body tissues. And COVID onset lung diseases also have similar impacts as they prevent oxygen from reaching your organs and deprive them of oxygen. So, further carbon monoxide may worsen this condition by preventing even more oxygen to reach organs. In the same way, sulfur dioxide has been related to many lung diseases. And like carbon monoxide, it could worsen the impact of COVID onset diseases such as ARDS, asthma, and chronic obstructive pulmonary disease. On the other hand, taking mobility into consideration, the model showed that an increase in residential mobility, in fact, reduced the amount of cases and deaths. An increase in residential mobility means more people are staying at home. This makes sense because COVID is spread from person to person via direct touch or respiratory droplets. So, this finding basically reinforces the fact that government policies such as lockdowns were effective in keeping the virus under control. And
Ishaan,
yeah,
I'll interrupt you. Thank you so much. If time is up, that was great, a great presentation, and I think you were in the summary section. So thank you so much for sharing all the learnings. Any questions for Ishaan? And I saw that Madhavi joined, and Madhavi is not a presenter. Madhavi, did you want to ask a question? All right. So Ishaan, I have a question for you. You mentioned that you got, well, actually, Andrew just asked a question. So Andrew is asking, have you considered that pollution causes an increase in cases before it shows an increase in non-residential mobility?
So I think that what I found that these two pollutants, carbon monoxide and sulfur dioxide, so I feel are majorly they majorly come from industries. So sulfur dioxide is found from agricultural industries and other types of industries. But at the same time, they're also found from vehicular emissions. So yes, to some extent, I do think that since residential mobility increased, the pollution from these pollutants were decreased. But but I feel that industries are major causes of these pollutants, and governments maybe should try and controlling these emissions as they might reduce COVID cases too.
Got it, Ishaan. One final question for you. What was the hardest part of the project?
So honestly, for me, the hardest part of the project was acquiring data itself. So there was a lot of incomplete data, and in fact, I for like 2 3 weeks I had to go requesting for pollution data from the Central Pollution Control Board until they finally gave it to me. So I
Got it.
felt that was my hardest
Yeah.
part.
Ishaan, thank you so much for the great presentation. Thank you.
Thank you.
So next up will be Sammith Singamsetty, from a student from Vidyaniketan Public School from Bengaluru, Karnataka, India. So Sammith, please join us. Hi, Sammith.
Hello, am I audible? Yeah.
Yeah, you are. So let me leave the page.
Yeah, just a second. Is my screen visible? Is my screen visible?
Yes, please go ahead.
Oh, okay. Sure. Sorry. Yeah. Hello, everyone. My name is Sammith Singamsetty, and today I'll be talking about my research project, which is mapping the dark matter halo of Andromeda Galaxy. So I have a timeline for my presentation. First, I'll talk about dark matter. Then I'll explain some key terms and concepts. And then finally, I'll talk about my research and about the results and limitations. And finally, I'll answer any questions that you have. So first, let's have a brief overview of dark matter. The most important question is, what is dark matter? Now, dark matter accounts for 22% of what is there in the universe. So the stars, the planets, the galaxies, everything that we see just accounts for 4% in the universe, the small orange slice that you see in the pie chart. So dark matter gets its name because it does not interact with light. And because we haven't detected it yet, we do not know what it is composed of. Another question that naturally arises is, why do we even need to have dark matter in the universe? So there are a lot of reasons, or there are a lot of reasons for the presence of dark matter, but the three most important ones are listed here. The second reason, which is to explain the rotation velocities of objects present in galaxies, is a central idea to my research project. Now let's move on to some of the terms and concepts we need to know. The first important concept is the dark matter halo. So immediately when you see the image, you can see that the dark matter halo extends far beyond the edges of the galaxy. And you can also observe that galaxies evolve inside dark matter halos. So this means that the properties of the galaxy are very closely related to the properties of the halo in which it resides in. So knowing things about the halo can actually help us understand the evolution and the history of the galaxies. The second concept, and the most important concept for my research project, are rotation curves. Rotation curves give us the rotational velocity of objects present at a certain radius. Now, this is the general formula for the rotation velocity, and this is obtained from the standard Newtonian model. The variables are also listed. In this formula, g is the universal gravitational constant, r is the radius at which the object is located at, and m is the mass that is contained within radius r. Now, to explain rotation velocities, I actually find it really helpful to take the solar system, which is a much scaled down version of a galaxy. Now, there are a lot of similarities between a galaxy and a solar system. The sun is located at the center of the solar system, and a black hole is located at the center of the galaxy. Now, I thought now we can map the rotational velocities of planets in the solar system, and this is represented in this graph. So you will see that Mercury, which is the closest planet to the sun, has the highest rotational velocity, and Neptune, which is farthest away from the sun, has the lowest rotational velocity. So this can be explained by using our velocity general formula for rotational velocity. If we consider the planet Mercury, it's the closest to the sun. But the mass that is contained within Mercury's orbit is the mass of the sun, and the mass of the sun accounts for 99.8% of the mass of the solar system. So this means that the mass that is contained within Mercury's orbit is essentially the mass of the solar system. So this means that Mercury will have the highest rotational velocity. But if we take Neptune, for example, the distance between Neptune and the sun is the highest. However, the mass that is present within Neptune's orbit is again the mass of the sun. but the radius has increased in this case. So this reduces the rotational velocity of Neptune. So this sort of mass distribution, where mass is mostly concentrated at the center, yields a rotation curve that slopes downward. So the objects that are present towards the center have a higher rotational velocity as compared to objects that are present farther away. So astrophysicists determined that, or they predicted that it would be the same for the galaxies. They predicted that the mass of the galaxy would mostly be at the center, and the rotational velocities would decrease as the distance increased from the center. But what we observe in nature is actually incredibly different from what we have predicted. We predicted decreasing orbital speeds with increasing radius because we estimated the mass of the galaxy to decrease as the radius increased. But what we actually observe is that there is an increasing orbital speed as the radius increases because the mass of the galaxy has increased. Now, this is extremely puzzling because our understanding of the mass distribution is wrong. But astrophysicists were really puzzled because they had taken into account all the luminous matter, the matter that they could see in the galaxy. But still, they were not able to produce the rotation curves. This meant that there is the presence of additional matter that we have not yet detected that is aiding to the rotation curves. So this was the aim of my research. In order to map the rotation curves of the Andromeda Galaxy. To do this, I utilized dark matter density profiles. Now, a brief description of dark matter density profiles. They give us the density of the galaxy at a certain radius by taking into account the dark matter component as well as the luminous matter. So there are 6 density profiles, and the most important variables are listed here. Now, you may be wondering, how would I get the rotational velocities from density profiles? So we know that in our formula for the velocity, the mass component is the one that needs to be changed. So we can get the mass component from a density profile by taking the integral of the density profile from 0 to a certain radius r. So this is what I did for my research. Also, I utilized 2 data sets, the Corbelli dataset and the Carignan dataset. Now I'll talk about some of the results and limitations of my research project. So these are the rotation curves for Corbelli dataset. And you can see that all the density profiles provide wonderful fits for the dataset. The first fit, which is the lucky 13 fit, it provided the best fit. And the pseudo-isothermal profile, it provided the least accurate fit. So this order was almost the same for the Carignan dataset as well. So the lucky 13 profile, it provided the best fit, whereas the pseudo-isothermal profile provided the least accurate fit. So there are, of course, limitations for my research project. The first limitation is that density profiles actually fail to give the density at smaller radii. And there are a lot of other issues with density profiles, but solutions have been proposed to curb these things. And the density profiles are extremely accurate now. The other problem has to do with data sets. So rotational velocities can be only obtained until a certain extent. And beyond that, our current technologies cannot pick it up. So finally, a few conclusions from my research project. The main and the most important conclusion is that the rotation curves provide evidence for the existence of dark matter, and the density profiles, they provide a way for us in order to prove the existence of dark matter. So finally, I come to the end of my presentation. Clear skies, as any amateur astronomer would say. A huge thank you to Polygence for giving me this wonderful opportunity to pursue a research project in high school. And a huge thank you to my mentor, Nicholas Lapidus, for his countless support and contributions.
Thank you. Thank you, Sammith, for that presentation. And I think we have time for one question. Well, I'm a physicist, I'm proud. And so I'll just ask you, how do you think people gather the data?
Could you repeat the question? I couldn't hear you.
Yeah, so if you could mute my while I ask this question, that would be helpful. Wonderful. So do you know how we gather all this data about distant galaxies?
I'm not very sure about that. Would it be the light emitted from the visible hydrogen, the 21 centimeter line, as well as the light emitted from the objects? I would guess from those 2 sources is how we obtain it.
That's certainly one of them. Alok, do you have a question? Let's bring in Alok.
Can you hear me?
Yeah.
Great presentation, Sammith. I really learned a lot about dark matter. One of the questions I had was about your formula for rotational velocity with the square root of how come the r's don't cancel in the numerator and denominator?
Okay, yeah. So I'll just share my screen for that so that it becomes clear. Yeah. So in my formula,
we have 1 minute, so yeah.
Oh yeah, I should do that. So in this formula, actually, the r doesn't cancel because I am writing the mass as a function of the radius. So the radius is a individual component, and the mass is just given as a function of the radius. So I'm talking about the mass which is contained within a certain radius, and the radius itself is a different component. So that's why they don't cancel out in this formula.
Thank you. Thank you so much, Sammith, for that presentation and the wonderful work. And next up, we have Jacob Rojit, who is from Delhi Public School, Bangalore South, from Bangalore, Karnataka, India. So, Jacob, please join the stage. We are waiting for Jacob to join. Hi, Jacob.
Hey.
Please start sharing your screen. We are excited to get started with your presentation.
Is it visible?
Yes, it is. So Jacob, please feel free to get started.
Cool. Hello everyone, I'm Jacob Rojit, and today I will be talking about how me and my mentor Bryce developed a model to analyze the impact of carbon dioxide emissions on global temperature change. So a little bit about me. I'm from Bangalore, India, and currently a senior at Delhi Public School. I enjoy studying environmental science and chemistry, and while I'm not studying, you would usually find me playing soccer or listening to music. Now back to the project. So as we all know, the Earth has been warming considerably over the past few years, with this warming being caused by human interference and a little bit of natural variability, with the preponderance of evidence showing that humans are mostly responsible for the Earth's warming. 2/3 of global warming has occurred after the year 1975, with the Earth warming at an average rate of 0.15 degrees Celsius to 0.2 degrees Celsius per decade. This global warming has led to what we know today as climate change. Climate change has increased the frequency of multiple natural disasters, such as forest fires, floods, droughts, frequent flooding, untimely snowfall, hurricanes, and so much more. The graph over here is from the climate report of the IPCC, the Intergovernmental Panel on Climate Change. According to this report, in the year 2017, human-induced warming resulted in a temperature increase of 1 degree Celsius when compared to temperatures of the pre-industrial times. The report also says that if we were to continue at the current rate of warming, then the temperature of the planet would increase by 1.5 degrees Celsius by the year 2040. So what does our project do? Our model analyzes how reduction in carbon dioxide emissions impacts temperature change. Specifically, we focus on what proportions we must decrease the use of fossil fuels to limit warming to around 1.5 degrees Celsius by the year 2100. For this study, we have identified coal, oil, and natural gas as the fossil fuels that are major emitters of carbon dioxide. We then model the impact of replacing these fossil fuels with clean, renewable, and non-emitting sources of energy. For our project, we took data from the NASA and NOAA database. This was temperature anomaly data with respect to the base temperature period of 1951 to 1980. And for our project, we adjusted it to the years 1880 to 1900. Conversion metrics for converting energy produced into carbon dioxide emitted was taken from the US Energy Information Administration and the US Environmental Protection Agency. And data on carbon dioxide emitted and the use of each fossil fuel was taken from the 70th edition of the World Statistics Energy Report published by British Petroleum. So when we started our project, we did not have data for the carbon dioxide emissions of each fossil fuel. So in order to figure out how much carbon dioxide was emitted by each of our fossil fuels, we used the metrics published by the EIA and the EPA. This gave us CO2 emitted for each of the fossil fuels, which we then graphed against year of observation and derived equations to predict the amount of carbon dioxide that would be emitted over the next few years. Now that we had our carbon dioxide emission, the next step was to verify whether coal, oil, and natural gas are the main emitters of carbon dioxide. In order to do this, we compared calculated carbon dioxide emissions and graphed it against recorded CO2 emissions for the years 1981 to 2014. The scatter plot over here gives us the line of best fit with an R squared of 0.994, which confirms that our hypothesis that coal, oil, and natural gas are the major emitters of carbon dioxide is correct. Once we had confirmed that our fossil fuels that we were studying are the main emitters of CO2, we needed to develop an equation to predict how temperature would change over the next few years. In order to do this, we graphed temperature anomalies for the years 1981 to 2014 with respect to the base period of 1880 to 1900. This temperature anomaly was graphed
Jacob, are you here? Let's wait for Jacob to rejoin. Can anyone indicate if they can hear Jacob? Got it. So obviously, this is both the beauty as well as the downside of a virtual conference. We are joining this session from all over the world, which means that we are at the mercy of Wi-Fi issues. But we did budget a little bit of extra time, so we can, make sure that we can hear Jacob out. So hopefully Jacob will be able to rejoin in a moment. Here is Jacob. Hey, Jacob, welcome back.
Yeah.
Yeah. And continue. We are super, you built up that tension. We were just waiting to see what the next slide would be.
Yeah, so I think I was over here. Yep. So yeah, once we had our equation for the change in temperature, we used it to predict the amount of temperature change for the year of observation, and this was done by plugging in the carbon dioxide emission value for the given year of observation. We used this equation to analyze multiple scenarios and then narrowed it down to 5 scenarios to give you a broad picture of what would happen. Our scenarios include reducing the use of coal by each year, reducing the use of each fossil fuel each year, and what would happen if there is no change. We chose coal over oil and natural gas because according to our graphs, coal is what would be used the most over the next few years, also resulting in coal having the highest carbon dioxide emissions amongst the 3 fossil fuels. So the dotted line on our graph shows the year 2014. I'm going to start with explaining the black line. The black line shows what would happen if there is if we do not change the use of our fossil fuels over the next few years. This would lead to Earth warming by 2.55 degrees Celsius when compared to pre-industrial times. And according to the IPCC, this would be a point of irreversible climate change. The green line on your graphs shows what would happen if we were to reduce the use of each fossil fuel by 3% each year. That would result in temperatures returning to somewhere around the same as what it was during the pre-industrial times. The red line on our graph shows what would happen if there was a decrease in use of fossil fuels by 0.5% each year, while the blue line shows what will happen if there's a decrease in the use of coal by 3% each year. The next line is the orange line that shows us the outcomes if there would be a reduction in the use of coal by 1% each year, and that is what we will be talking about now. The orange line, which shows reducing coal by 1% each year, shows us that if we were to reduce coal at this rate, it would result in the Earth warming by 1.52 degrees Celsius, which, according to the IPCC, would be a temperature level at which climate change would not be irreversible. So that would mean if we were to reduce the use of our fossil fuels and replace them with renewable and non-emitting sources of energy, it would be possible to reduce the uses of climate change, which could in turn prevent the occurrence of disasters such as floods and forest fires more frequently. Thank you. And I'd like to give a quick shout out to my mentor, Bryce, for his guidance and support throughout my project. I couldn't have asked for a better mentor. And I'd like to thank Polygence for matching me with Bryce and giving me the opportunity to be here today. And thank you, everyone, for listening to me. We will now be moving on to questions.
Thank you, Jacob. So Andrew already has his question typed out, and I'll read it. When you tested what would happen if we reuse coal use only, what did you assume would happen to the other fossil fuels? Did you say they would stay the same as baseline levels, or did you extrapolate an increase based on data over the last few years?
So when I assumed that, I'm sorry. So when I assumed that there would be a decrease in coal, I said that I factored in a continuous increase for the other two fossil fuels, oil and natural gas. So I did not leave them at baseline levels. I let them increase over the next few years.
Got it. Thank you. Thank you from Andrew. Any other questions? Bryce says.
Thank you so much, Bryce. Thank you.
Anyone else wants to ask a question? Feel free to ask to share your camera and audio and ask the question live. So, Jacob, let me ask you, what was the hardest part of the project?
The hardest part was trying to figure out how we were to relate fossil fuels to carbon dioxide emissions to temperature change, because there weren't official metrics and things like that. So we had to do a lot of matching and moving things around. And in the end, we were pretty happy with the results we had.
Wonderful. Well, thank you for the present log. We have 1 minute and then we need to move on. So.
Hey, great presentation, Jacob. Quick question for you. Just like, thinking in the other way, the Earth goes through periods of cooling and heating in the long scheme of things. Have you thought that, your data set is only a small fraction of the overall length of human species on Earth, as well as Earth being around, for billions of years, and that we have like periods of warming and cooling, that we're right now maybe going through a period of warming. And this is probably not a, globalized situation, like in terms of all the variables, but rather just local to this time period.
Jacob, are you here? Well, I think we might have lost Jacob and we need to move on. So Jacob, please answer that question by Alok in the chat. And please feel free to connect. And we will move on to Tyler Kuo, a student in Basis Scottsdale Charter School from Scottsdale, Arizona, the United States. So Tyler, please join the stage and share your audio and video. Hey Tyler, please share your screen.
Hey, thank you. So let me get started. So, hi, I'm Tyler, and today I will be presenting on the role of stem cells in cancer therapies. So, first I just wanted to go through a quick overview. So first we're just going to go through like some cancer facts, then we're going to go through some types of cancer treatments, the side effects of these cancer treatments, and then how doctors deal with these side effects, some future applications, and then a discussion. So first, let's just start with cancer. So I'm sure we all know, but cancer is a really big problem in today's current society. So for example, in the United States in 2021, it was predicted that 1.9 million people will be diagnosed with cancer and 600,000 of them will die. And not only that, but 1 in 2 women and 1 in 3 men are likely to get cancer throughout their lifetimes. But, I don't want to be a downer because since the advent of new cancer therapies, the cancer death rate has actually been steadily decreasing. And this is largely due to advancements in technologies like chemotherapy and radiotherapy. So I'm just going to go talk about those for a little bit. So first is chemotherapy, and chemotherapy, I'm sure all of us have heard about it, but I'll give you a refresher. So it's the use of drugs to kill rapidly growing cancer cells. And so as you can see in the image in the top right here, basically a patient is injected with an IV, and then the IVs basically give them the chemotherapy drugs. And so chemotherapy has its positives and negatives. So the advantages is that it basically works exactly as intended. So it can shrink or kill the tumor, and it makes surgery possible, right? But the disadvantage is that chemotherapy can lead to fatigue, dizziness, and it weakens the immune system. And also, how chemotherapy works is that it targets rapidly dividing cells. So in some cases, that can mean cancer, but in other cases, that means it can also harm healthy, rapidly dividing cells. So this is why people who receive chemotherapy often lose their hair, because hair divides rapidly, so the chemotherapy drugs basically suppresses it. But it's not only limited to hair, it can also basically impact the bone marrow, because bone marrow also divides pretty rapidly. And so bone marrow is a really big part of my presentation, and I'll talk about it more later. But first, I wanted to just kind of go into radiotherapy really briefly. So radiotherapy is the other main type of cancer therapy. It's the use of intense energy to kill cancer cells. And as you can see in the top right here, this patient is put under some equipment, and basically they're given radiation. And as you can see in the bottom left, this radiation targets the tumor. So the advantages are pretty much the same as chemo, right? You're just trying to kill the cancer. And so you're trying to shrink or kill it. And the disadvantages are relatively the same too. The only difference is that instead of targeting rapidly dividing cells, radiotherapy targets one area. So as you can see in this kind of gross looking picture in the bottom right here, this is the patient's skin after he receives chemotherapy. As you can see, it's targeted all in one area. But then again, I do want to emphasize that bone marrow suppression is the most important one that I'm going to be talking about. And so I'll get into it on this next slide. So what does the bone marrow do? Well, the bone marrow actually houses hematopoietic stem cells. And so what a stem cell is it's a cell that can differentiate into more specialized cells in the human body. So basically, it can become like a muscle cell, a skin cell, a brain cell, or a blood cell. But hematopoietic stem cells, which are the ones inside the bone marrow, are basically can only become blood cells. And so when I'm talking about bone marrow suppression after chemotherapy and radiotherapy, I'm really talking about a depletion of hematopoietic stem cells. And so when you have a decrease in hematopoietic stem cells, that basically leads to less blood cell production. And this is really bad because it can lead to fatigue and other side effects like a weakened immune system. So what do scientists do to combat this problem? Well, they do something called stem cell transplants. So basically there's two types. The first one I'm going to be talking about is autologous stem cell transplants. And basically, this is when the patient's own hematopoietic stem cells are returned back to them. So as you can see in this diagram in the top here, basically you have a person with cancer, and before they receive chemotherapy or radiotherapy, a doctor takes hematopoietic stem cells out of their body and stores them for later. Then, after they receive this high-dose chemotherapy or radiotherapy and their bone marrow is suppressed, the doctors give the stem cells back to the patient. And basically this helps recovery a lot. And so it has its advantages and disadvantages. The main advantage of autologous stem cell transplants is that there's no risk of rejection. So basically, since you're returning a patient's own cells back to them, there's no risk of the body rejecting it because they know what their own cells look like. And so it's considered the safer one of the two types of transplants I'll be talking about. But the disadvantage is that there's a higher risk of relapse compared to allogeneic stem cell transplants, the other one I'll be talking about. And this is because basically you receive chemotherapy and radiotherapy, and these stem cells are returned back to the patient, right? But if there's any cancer cells remaining after the chemotherapy, these stem cells won't be able to recognize it, right? Because the body already hasn't recognized the cancer, so that's why you had to get chemotherapy. So these new cells won't recognize it either. So there is a higher risk of the cancer dividing and relapsing again into the patient. But for this other type of stem cell transplant, this isn't really a problem. So allogeneic stem cell transplants is the use of outside donor cells to replenish the bone marrow. So as you can see, it's kind of pretty much the same as autologous, except instead of the patient's own stem cells being used, it's a donor stem cells. And usually this donor is like a sister or a close family relative. And so these donor stem cells are given back to the patient. And it also has different advantages and disadvantages. The advantage is that there is a potential of graft versus malignancy effect. So what the graft is basically what is being given to the patient. So it has stem cells, but it also has stuff like T cells. And so basically what can happen is that these T cells cannot recognize, these new T cells can recognize the remaining cancer cells, and then they'll attack the cancer cells and eliminate them. And this reduces the risk of relapse. But on the other hand, there's a risk of graft versus host disease, which is basically the other side of this. So instead of the T cells not recognizing the cancer cells, it doesn't recognize the healthy patient cells, and so it attacks the healthy cells. And so now I just have a few clinical applications. I'll just go through them really quickly because of time. So there's lymphoma and multiple myeloma, and these are good examples of autologous stem cell transplants. As you can see in this figure in the right here, the tumor has been significantly reduced after chemotherapy, and although it doesn't show it in the picture, the autologous stem cell transplant really helped with recovery. And then there's also allogeneic stem cell transplants. It's shown here in acute myeloid leukemia. Acute myeloid leukemia has a decently high risk of relapse, so allogeneic stem cell transplants really help with the graft versus malignancy effect in this case. And then the final thing I kind of wanted to go through was just a future application that I thought was pretty cool. It's called CAR T therapy. And so this is a type of immunotherapy. So basically the immune system does all the work, and the T cell receptors are engineered to cause these patient T cells to attack a specific protein. So how it works is you have this patient with cancer, as you can see in the bottom here, and their T cells don't recognize the cancer. So the doctor takes the T cells out of the patient and basically engineers them in a way where the cancer can now recognize the T cell. And so, the T cell cannot now recognize the cancer. And so once you give back these T cells, they start to attack the cancer and basically eliminate the cancer from the body. So I thought that was pretty cool. And so, yeah, that's pretty much my presentation. In summary, there's a lot of cancer therapies like chemotherapy and radiotherapy, but they have their downsides like bone marrow suppression, which is why they require these stem cell transplants. And so we went over the types of stem cell transplants and their future applications. So that pretty much concludes it for my presentation. I'm now open for questions.
Thank you, Tyler, for the great presentation. Questions from the audience? Feel free to request joining the stage so we can ask it live or type it into the chat. All right, in the meantime, so Tyler, you obviously covered some ground here. Which part of this research was the most challenging for you?
It was definitely, I would say, finding clinical applications for allogeneic stem cell transplants, because it's kind of hard because allogeneic stem cell transplants have a lot of downsides and they're a lot riskier than autologous. So it's hard finding some examples to use for that.
And how do you find them?
Well, I just looked into a bunch like Google Scholar. Usually they have like clinical applications, but it was kind of deep. It was like on Google page 3, so I had to read through a bunch of research articles to find it.
Got it, Tyler. Any other questions? We have 1 minute for questions. Well, Tyler, thank you very much for your presentation. And up next will be Tanisha Joshi from Dublin High School from Dublin, California. So Tanisha, please join the stage.
Okay. Can you hear me?
Got it. So Tanisha, we just had one quick question about for Tyler. So Tyler, please respond via chat. And Tanisha, please feel free to share your screen and start your presentation.
Okay. Can you guys see it?
Yes, we can. Please start.
Okay. So hi, I'm Tanisha Joshi, and I will be reviewing theories of neurodevelopmental disorders through organoids. As of now, our knowledge regarding neurodevelopmental disorders is limited, consequently decreasing treatment efficacy. So by increasing public awareness of ASD and emphasizing the urgency to research contribution, we can advance therapies for neurodevelopmental disorders. As there's no current medical treatment for ASD either, it's important to model the disorder to understand how ASD functions and impacts patients in a mental and physical way. In this presentation, I will go over background information about important brain functions that impact neural circuitry to give insight about how errors in those pathways can lead to ASD, including concepts such as plasticity and how regular and specific receptors work in the brain. I will also describe the history of diagnosing and treating ASD while discussing current theories of how and why ASD occurs through various internal systematic functions, while reviewing the significance of the development of organoids and how they can be leveraged to model ASD. So in a healthy brain, electrical stimulation is required in order for a neuron or a nerve cell to fire. A neuron consists of many parts. The dendrites of a neuron are responsible for receiving information in the form of electrical stimulation. The cell body is responsible for processing the signal, while the axon helps transport the processed information across the neuron, and the axon terminals will send information to the next cell. This overall phenomenon is called an action potential, which enables communication in the brain in order to send commands throughout the body. The place where two polarized neurons meet to transfer this action is called the synapse, in which synaptic plasticity relates to the change in the strength of the synaptic connection between the neurons and its ability to change when facing alterations or challenges in the pathway of the cell, in which there are two types, chemical and electrical synapses. So famous neuropsychologist Donald Hebb was a prominent figure in the field of neuropsychology and was the founder of a principle called Hebb's Law, which concluded that neurons that fire together wire together, meaning that synaptic connection can continue to straighten the more a presynaptic repeatedly fires. So receptors mediate synaptic functions, especially NMDA receptors and AMPA receptors. These receptors regulate excitatory synapses and help neurotransmitters and messengers release signals from a presynaptic neuron, which increases the chances of an action potential in the postsynaptic neuron. So depending on the protein the NMDA receptor binds to, the process of LTD, or long-term depression of a neuron, occurs, or which is long-lasting decrease in synaptic activity, or LTP, which is long-term potentiation, or long-lasting increase in synaptic activity occurs. So many theories in neuroscience are often tested in mice, which are used as model organisms because they share similar biological characteristics with humans. For example, mice and human genomes have similar proteins while sharing similar functions in organ regulation. And this reflects the concept of translation, which means that if a treatment successfully works in a mouse, then it should successfully work in a human because of their similar biological compositions. But however, through clinical outcomes, scientists have mentioned that there needs to be an improvement in the usage of mice as models, as there isn't enough data regarding neuropsychiatric diseases in mice itself, along with pre-existing diseases in mice as well. And this is due to the fact that success rates in translation is low in experiments, and therefore organoids present a unique advantage, which is reducing the usage of mice in research, along with experimenting with human-derived culture tissue to model human-specific neurobiological disorders while reducing the usage of mice in experiments as well. So through research and assessments, doctors have correlated repetitive motion and inflexible communication as symptoms of autism, and noticed that patients with the neurodevelopmental disorder are biologically different in terms of brain structure compared to people who do not have autism. Autism was also discovered to be a heterogeneous disorder, meaning that both genetics and environmental risks play a role in its occurrence, and having a detailed history of a patient would be beneficial during the diagnosis procedure. So in this case, various tests help confirm diagnosing autism in patients, the two main being behavioral and technological tests. So EEG are used to determine the level of brain function used and to show how synaptic changes occur in the brain. Another machine is called an MRI machine, which helps doctors understand how the brain is functionally and structurally different due to disrupted pathways in the neural circuit. And through behavioral diagnosis procedures, the Early Start Denver model is used is an autism management tool to observe, while the PECS system, or the Picture Exchange Communication System, aids in speech generation with training and behavior-related tasks. Drugs and medicines aren't normally used to specifically care for autism, but a few are used to manage out of control or severe behavioral problems. In a properly working neural system, excitation and inhibition are signaling processes that regulate neuronal firing. However, sensory dysfunctions can impair EI balance, or excitation and inhibition, within a network of neurons. People who also have sensory processing dysfunction are often overly sensitive to objects and affect the way the brain processes sensory information. And dysfunctions like these correlate with brain developmental issues. So somatosensation is also affected by natural biological processes in the human body through interconnected pathways. The gut microbiota brain axis is a pathway utilized by the brain to help the gut communicate with the central nervous system in a bidirectional method through indirect and direct signaling with proteins. The gut microbiota communicates with the CNS through the vagus nerve, which helps transmit information to different organs from the body to the brain. And the vagus nerve uses mechanoreceptors that are triggered by hormones to begin carrying out its functions. So disruptions in the gut microbiota through bacteria or other cells causes neurodevelopmental disorders, such as ASD, if homeostasis isn't regulated, which can lead to problems like the failure of proper brain development. As of now, there's a lack of effective methods that are used to treat or minimize the effects of oversensitivity that's caused by sensory or somatosensory dysfunction. So specific genes such as SHANK3 or MECP2 are correlated to ASD, as many neurodevelopmental diseases are often gene-related. Loss or overexpression of these specific genes correlate with ASD. An experiment was conducted in mutant mice with ASD, in which they were exposed to the chronic treatment of ASD genes as a way to improve their behavior and social impairments. And as a result, the scientists did in fact see improvements in the behavior of the mice through reducing their anxiety and fixing some of their abnormalities in their brain as well. So using the process of translations, as we previously talked before, this treatment could be effective in calming some of the effects of sensory dysfunctions in humans. So scientists have used different types of models in their experiments, primarily animals. For example, action potential and its generation were originally discovered through squids and their axons and electrode mechanisms. Similarly, the frog was used to study synaptic transmission because of their behavior, and the brain synapses were involved, while the rat was used to study spatial representation because of their behavior and actions. But mice are most common, as stated before, because of their biologically similarity to humans. However, as technology continues to increase and develop, there's more ethical and advanced options to model research regarding neuroscience and neurodevelopmental processes or diseases. Organoids, or replicas of organs that are grown via cultured stem cells, are revolutionized as being the next way to display and model different diseases. Through the utilization of organoids, various neurodevelopmental disorders can be modeled through the stem cells of the diseased tissue and be a way to model other polygenic or monogenic diseases. Stem cells are cells that have been collected from the original organ but are used to develop organoids, which could be considered as a replica. And organoid technology has grown over the past few years, leading to in vitro studies of disorders in brains that are in embryos. And reconstructing organs through organoids requires a vast amount of technology in different branches of science, including transcriptomics, proteomics, and epigenomics, which are studies of genomes, utilization of cells, and correlating genetics and behavior. All of these branches have an important role in understanding how organoid functions and how they would manage to replicate an actual organ using derived stem cells to grow and nurture stem cell tissue. So CRISPR is a method of genetic alteration and is a tool used in reconstruction of organoids.
I'm sorry to interrupt. Time is up, so if you could quickly summarize, that would be most helpful.
Okay, yeah. So basically, CRISPR allows editing and alterations of genome sequences, and they could also be used to correct these genes incorporated in diseases and model the specific diseases as well. And scientists have been successful in experimenting with CRISPR in autism-related testing. So in this case, psychiatrists and doctors have researched autism over the past few decades, and discoveries have been made. But despite the achievements, there has been much to be learned about autism in terms of establishing a permanent solution and utilizing technology while doing so. So as technology continues to evolve, researchers can broaden their horizons in the neurobiology field as well. So I just wanted to thank Polygence for giving me this opportunity to research and present this topic. And I also wanted to thank my mentor, Sonika, who was very supportive and helpful throughout this whole procedure.
Thank you, Tanisha, for that presentation. We have about a minute for questions. Feel free to ask in the chat or join the stage. So Tanisha, while others are gearing up, this story about organoids, I haven't heard them before. I'm not a biologist, I'm a physicist. So these are, how do you create these environments? I was a little confused there.
Yeah, so organoids basically are, you have to have like a specific type of procedure in order to grow them. So obviously like people use like incubators or other different types of machine in order to grow the cultured tissue, because it's not like a natural process, you know? So in that case, you would need like the specific machinery and tools in order to grow the specific tissue. So in that case, people use incubators or other types of machines to do so.
Got it. Any other question? So one very quick question. What was the most challenging part of the project?
I would say that finding experiments that correlated through the use of organoids, like using organoids to treat ASD or experiment in like the ASD related field, because overall the organoid field is pretty new, and CRISPR as well. So finding specific experiments that related to the topic that I was interested in was kind of hard, since it's a new topic and they're still working on it. But there were a few, so I was able to find them.
So thank you so much for your presentation and your hard work. And for those who are still here amongst the presenters, please join the stage for a moment so we can all applaud you quickly for the hard work and amazing presentations you have put together. Tyler is joining. Ishaan is joining. All right. So thank you so much again. Sammith is joining. So please mute your microphone while I just say a few words. So, you know, for me, it's just incredible to see all the hard work that you've done and the great job you did with the presentations. And for me, this is really the most inspiring thing about Polygence, that you guys are so dedicated and inspired to do this hard work. And I think that opening up your minds and being curious and doing all this work really bears fruits down the road. And learning for learning's sake is where I think the biggest acceleration comes when it comes to your academic career. So huge clap to you. And from the audience as well, we got a lot of questions, which shows that your presentations were very intriguing for everyone. So I just really wanted to take a moment to reflect on your journey thus far and say, please keep up this great work and enjoy your research. And you know, things get hard when it comes to academia, but eventually you can push through. And thank you again. So a big round of applause for those of you listening in the background, to Tanisha, Tyler, Ishaan, Sammith, and Jacob. And please feel free to connect with our speakers. They have a lot more to share. They only had a limited amount of time. Unfortunately, I had to cut off some of the exciting stuff that was still to be shared. So reach out to them, congratulate them, make sure that your questions are answered. And with that, I think we should all jump to our keynote speaker's presentation. We have Professor Margot Gritson from Stanford, Professor of Data Science, presenting to us in just 1 minute. So please head back to the reception area and choose the keynote speaker and join there, and we'll see you there. Great job again. See you there. Have a good one. Bye.